So if you walk into your bathroom right now and just, open the medicine cabinet, you probably see a row of little plastic bottles.
Right, yeah. Vitamins, painkillers, all of that.
Exactly. And in our minds, those bottles sit in this very specific secure category. Like they are safe, they are the
Oh,
healers.
totally. We trust them completely.
Right. But then you look in the cabinet under the sink, where you keep the bleach and the pest control sprays, and that is the danger zone. That is where the poisons live.
Yeah. We like to keep those worlds very separate.
It feels like this very clean binary system. But today, we are taking a deep dive into the language and the science of medicine to just, well, completely shatter that comforting wall.
We really are.
Well,
Because
good.
it is a very comforting way to view the world, honestly. We desperately want this solid, impenetrable wall between the things that save our lives and the things that, end them.
Yeah, but we're going to look at how the words we use for healing and the words we use for killing are actually identical. We're exploring the surprisingly, almost terrifyingly blurry line between a life-saving drug and a deadly poison.
And to really see how blurry that line is, we just need to look at three foundational pieces of medical terminology. Things you'd encounter in, like, the very first week of any baseline pharmacology course.
Okay, lay them out for us.
So on paper, they look completely distinct. First, you have pharmaco, which is the combining form for drug or medicine, so think pharmacy.
Right.
Then you have toxo, which means poison, as in toxicology.
Makes sense.
And finally, you have this suffix, sacchalytic, which means pertaining to loosening, dissolving, or breaking down.
Okay, so on the surface, they look like three totally separate tools in a toolbox. You have one word for your medicine cabinet, one word for the poison under the sink, and one word for, like, a chemical action.
Yeah, exactly. But here is the massive twist that most people never realize. Pharmacology and toxicology are not two different subjects.
Wait, really?
Really, they don't study two different kinds of substances. They're the exact same discipline.
Yeah.
They study the exact same substances.
That is just, wow.
Yeah, the only difference between them, literally the only difference, is the dose and the context.
Which, if you're anything like me, hearing that for the first time breaks your brain a little bit. Because to really wrap our heads around how these two fields are essentially twins, we have to look backwards at the language itself.
We do, yeah.
And looking through our notes on the history here, these two totally different concepts, healing, drug, and deadly poison, they actually used to be the exact same word.
They were. If we travel back to ancient Greece, we find the root of both of our modern fields in this single, highly ambiguous word, and that word is pharmakon.
Pharmacology.
Right. The ancient Greeks did not linguistically separate a healing substance from a harmful one. Pharmakon meant drug, remedy, medicine, and poison, all simultaneously.
Which is just wild for modern minds to process. It's like having the exact same word for, I don't know, a life raft and an anvil. You wouldn't know if it's going to save you or sink you until you actually throw it in the water.
That is a perfect analogy.
It's so ambiguous that the 20th century philosopher Jacques Derrida actually wrote an entire essay called Plato's Pharmacy just about how you cannot cleanly translate this word. Because it is structurally a cure and a poison at the exact same time.
And how that single concept fractured into the separate words we use today is just a fascinating linguistic evolution. So you have pharmakon covering the entire spectrum of substances, right?
Right, both the good and the bad.
So you might wonder, where does our modern word toxic actually come from?
Yeah, because toxo means poison, right?
Well, it actually comes from a much narrower Greek phrase, toxikon pharmakon.
And this is the part that I still, can't quite get over.
It's incredible. Toxikon pharmakon literally translates to poison for the bow.
For the bow, like a bow and arrow.
Exactly. It derives from toxin, meaning the weapon itself. Scythian archers and other ancient warriors would coat their arrowheads in venom or, plant extracts.
Oh, wow.
And over centuries, the phrase for this poison arrow just contracted. People dropped the pharmakon part entirely, and it just became toxikon, which moved into Latin as toxicum, and that ultimately gave us words like toxic and toxin.
So etymologically, at its very root, the word toxic doesn't mean poison at all. It means arrow.
Exactly. We named the poison after the delivery mechanism.
That is amazing.
Right.
Okay, so the Greeks were structurally ambiguous with their language, but eventually, we had to draw a scientific line between the cure and the killer. When did science actually step in and formalize this?
It actually took quite a while. Toxicology as a formal discipline is usually dated to 1814.
Oh, so pretty recent in the grand scheme of things.
Yeah, relatively speaking. It started with a Spanish-born chemist in Paris named Mathieu Orfila. He's widely considered the father of toxicology because he published the first systematic treatise that actually organized poisons by their chemical class and their physiological effect.
So he kind of brought order to the chaos.
He did. He moved the entire field away from, like, folklore and into rigorous science. And he actually pioneered forensic toxicology.
Oh, really? Like CSI stuff?
Basically, he developed methods to detect arsenic in exhumed bodies, which meant for the very first time, people could reliably be prosecuted for poisoning someone.
Wow. And pharmacology as its own distinct experimental science came a bit later, right?
Yes, that is usually credited to Oswald Schmiedeberg in 1872. He founded the first dedicated pharmacology institute in Strasbourg.
Okay, so that's the late 1800s.
Right, and he trained over 100 future professors there, who then spread that experimental discipline globally.
But looking at the sources, the real hero of our deep dive, the guy who built the actual conceptual bridge uniting these two sciences, he showed up like 3 centuries before either of them.
Yes, he did.
A Swiss physician named Paracelsus.
Paracelsus is a monumental, almost mythic figure in the history of medicine. So in the 16th century, he was actively rebelling against the rigid medical dogma of his time.
What was the dogma back then?
Well, back then, substances were categorized by fixed, inherent moral characters, like things were intrinsically hot or cold or, evil.
Oh, gotcha. Very unscientific.
Exactly.
Oh.
And Paracelsus completely rejected that. He insisted that toxicity is quantitative. It's just math. And he coined the ultimate rule that every modern toxicology course still opens with today.
Sola dosis facit venenum.
Yes, the dose alone makes the poison.
The dose makes the poison.
That is the organizing principle of both fields, the dose-response relationship.
So how does that actually work?
Well, every single substance on Earth, if you plot it on a graph of dose versus effect, produces a curve. If you have too little of a substance, it does absolutely nothing.
Right.
If you increase the amount, you hit the therapeutic window, and that's pharmacology, where the drug provides a biological benefit. But if you keep increasing the dose, you cross the threshold where it produces harm. And that's toxicology. It is the exact same curve for everything.
Come on, everything. You're saying the things we are literally made of, the things we need to survive, are toxins.
I am. Modern toxicology defines itself broadly as the study of adverse effects on living organisms. And that includes water, oxygen, and salt.
Water? Seriously?
Yes. All of those are toxic in excess. Think about it. If you drink too much water too quickly, your kidneys simply can't flush it out fast enough.
Okay.
The sodium in your blood drops perilously low, which causes water to rush into your cells. This causes swelling in the brain, a condition called hyponatremia, and it can be rapidly fatal. So yes, water is a lethal toxin at the right dose.
It really fundamentally reframes how you look at the physical world. And there are specific numbers that scientists use to map this out, right? We aren't just guessing where that toxic threshold is.
No, not at all. We have highly specific metrics. 2 key terms bridge these fields mathematically. The first is the therapeutic index.
And what exactly is that?
That is simply a ratio, just a single number, comparing the effective dose of a drug to the toxic dose.
Okay, so like a safety margin.
Exactly. A high therapeutic index means you have a lot of wiggle room.
Yeah.
You'd have to take way more than the normal dose to get sick. But a low one means the therapeutic dose and the toxic dose are terrifyingly close to each other.
And the second metric?
The LD50, which stands for lethal dose 50.
Okay, that sounds intense.
It is. It's the exact dose of a substance that would be lethal to 50% of a test population. It's how we standardize risk across every chemical on the planet.
Okay, theory is great, but I want to see this math in the real world. Because looking at a graph is one thing, but seeing a molecule flip from a healer to a killer in a human body is where it gets incredibly visceral.
It really does.
Let's look at something you might actually find in an English garden. Digitalis, from the purple foxglove plant.
Ah, yes. This is a beautiful historical example. So, in 1785, an English physician named William Withering published a detailed account of the foxglove plant.
What were they using it for?
It had been used in local folk medicine for a condition called dropsy.
Dropsy.
Yeah, today we know dropsy as severe fluid retention caused by congestive heart failure. Patients would essentially be drowning in their own fluid because their hearts just couldn't pump effectively.
That sounds awful.
It was. But Withering realized that if you extracted the active compounds, which we now call digoxin, and you dosed them perfectly, it forced the heart muscle to pump much harder, which cleared the fluid.
And we still prescribe that today.
We do. But Digitalis has an infamously narrow therapeutic index.
So not a lot of wiggle room?
Very little. It works by increasing intracellular calcium in the heart muscle. But if you overshoot that dose by even a fraction, the heart becomes dangerously overexcitable.
What does that look like for the patient?
Digitalis toxicity causes severe arrhythmias, nausea, and this very specific, bizarre neurological symptom. Patients start seeing vivid yellow-green halos around lights.
Which, as a quick art history aside, experts actually suspect might have influenced Vincent van Gogh.
Yes, I love this theory.
Right, because he was treated with digitalis by his physician, Dr. Gachet. So those distinct swirling yellow halos you see in his late paintings, like, the Starry Night, that might literally be him painting his own digitalis toxicity.
It's a fascinating theory.
Yeah.
And it perfectly visualizes that biochemical knife's edge we're talking about.
Totally.
Now, if we want to talk about the extreme end of the LD50 spectrum, we have to look at the ultimate modern pharmacon, and that is botulinum toxin.
Oh, this one is just wild.
It really is. Produced by a bacterium, botulinum toxin is by weight the most lethal toxin known to biological science.
So most lethal?
By far. We are talking about an LD50 measured in tiny nanograms per kilogram of body weight. It causes botulism by blocking the release of a neurotransmitter called acetylcholine at your neuromuscular junctions.
What does that mean in plain English?
Simply put, it completely paralyzes the communication between your nerves and your muscles. You suffocate because your diaphragm physically cannot move.
Oh my God.
Yeah. It's estimated that just a few hundred grams of this stuff, if evenly distributed, could literally wipe out the entire human population.
But wait, people literally pay hundreds of dollars to have this injected directly into their faces.
Exactly.
Botox. I mean, it is the absolute purest example of the dose-response principle in action.
It really is.
We take the deadliest, paralyzing poison known to humanity. We dilute it to an almost incomprehensibly microscopic level, and we use it locally. We paralyze tiny facial muscles to treat wrinkles, or, we use it to quiet hyperactive sweat glands, or even relax the muscle tension that causes chronic migraines.
And it highlights that the identity of the molecule hasn't changed at all. It's the exact same substance. The only thing that changed is the dose and the route of administration.
Right.
Which leads us to a third molecule, one with perhaps the most dramatic origin story of all, warfarin.
The rat poison that currently lives in millions of grandma's medicine cabinets.
That's the one. So back in the 1920s in the American Midwest and Canada, farmers were dealing with this devastating mystery.
Oh, what was happening?
Their cattle were spontaneously bleeding to death from, minor scratches or even internally for no obvious reason. And it turned out they were eating spoiled sweet clover hay.
How does clover cause fatal hemorrhaging?
Well, it turns out the spoiled clover contained a compound that essentially sabotages the liver. See, your liver needs vitamin K to manufacture the proteins that make your blood clot. And this compound completely blocks that process. Yeah. So a biochemist named Carl Paul Link spent years trying to isolate this specific anti-clotting agent. And in 1948, his lab finally synthesized a highly potent version of it. They named it warfarin, after the Wisconsin Alumni Research Foundation, WARF, which funded the research.
Okay, so they figure out what it is. Then what?
Because it caused fatal internal bleeding so incredibly efficiently, they marketed it exclusively as a highly effective rat poison.
But today, if you know someone who has survived a stroke or, has a mechanical heart valve, there is a huge chance they take warfarin every single day.
Right. Because within a few years of its release as a pesticide, physicians realized that the exact same vitamin K antagonism, just at a heavily monitored, much lower dose, was incredibly useful in human medicine.
Right, because it stops clots?
Exactly. It could prevent dangerous blood clots from forming in the first place. So a molecule specifically engineered and sold to kill rodents became one of the most widely prescribed daily life-saving blood thinners in the world.
It's just mind-blowing. So we spent a lot of time talking about the amount of the drug, the dose. But what about what the drug is actually doing to you?
The mechanism.
Right. Let's talk about the mechanics and clear up some major misunderstandings by returning to that suffix we mentioned at the very beginning, the fucklytic suffix. Because people hear words ending in a fucklytic and they immediately assume something bad is happening.
And this is a crucial distinction. Mechanics are entirely separate from morals. The suffix lytic just means breaking apart or dissolving. It has absolutely no moral valence on its own.
Right, like if we look at a word like hemolytic.
Exactly. Hemolytic anemia is a disease state where your red blood cells are being pathologically broken down and destroyed.
Which is bad.
Very bad. That is obviously a toxic outcome for the patient. But a thrombolytic therapy is a drug deliberately administered in an ER to dissolve a life-threatening blood clot.
Which is good.
Exactly. That is a highly beneficial outcome. The suffix lytic only tells you that dissolution is happening. Only the clinical context tells you if it's therapeutic or toxic.
So how does aelytic differ from other medical suffixes, like when we talk about antibacterial drugs?
I like to use an analogy here. Think of a bacterial cell like a locked house.
Yeah.
The suffixidal means killing. So a bactericidal drug is like poisoning the air inside the house. The physical structure is totally intact, but the threat inside is dead.
Okay, that makes sense.
Then you have the suffix static, which means inhibiting growth. A bacteriostatic drug is like putting a padlock on the door so the bacteria can't get out and multiply, but they aren't directly killed.
Got it. And syphilitic.
But syphilitic means physically dissolving or rupturing the structure. So a bacteriolytic drug is like hitting that house with a wrecking ball. It physically dismantles the bacterial cell wall. There are 3 distinct mechanisms of action.
That's a great way to picture it. But if the vocabulary is this rigorous and specific, where do people get confused? Like, what are the biggest public myths you see when it comes to pharmacology and toxicology?
Oh, definitely. The most persistent, dangerous myth is the appeal to nature.
Oh, yes.
The idea that natural inherently means safe and synthetic means toxic.
You see that literally everywhere in marketing today.
You do, and biologically, it is complete nonsense. I mean, we just talked about botulinum toxin.
Right.
It is 100% natural, produced by naturally occurring bacteria, and it is incredibly deadly.
Right.
Ricin, which is extracted from castor beans, is totally natural and highly lethal. Meanwhile, countless synthesized pharmaceuticals manufactured in steel vats have phenomenal safety profiles. The naturalness of a substance tells you absolutely nothing about its toxicity.
Okay, here's another one I see mix up all the time in the news. Toxin versus poison.
Right, so in precise toxicological terminology, they are not interchangeable at all.
Oh, they aren't.
No, a toxin is a poisonous substance produced specifically by a living organism. So snake venom, bacterial toxins, plant defenses, those are toxins.
Okay, and poison.
Poison is just the broader umbrella term for anything that causes harm. And to be really specific, a toxicant is the word used for human-made or environmental poisons like industrial chemicals or pesticides.
Okay, that's a really helpful distinction, and I think this brings up one of the most widespread misuses of toxicological language in, like, alternative medicine, which is homeopathy.
Yes.
Because homeopathy intentionally takes the dose-response math we've been talking about and throws it entirely out the window.
It does. Homeopathy is fascinating from a linguistic perspective because it superficially borrows the vocabulary of Paracelsus, but it completely inverts the logic.
How so?
The foundational practice involves diluting a substance, repeatedly shaking it in water over and over again until there are essentially 0 molecules of the original active ingredient left.
But they claim that extreme dilution actually makes the remedy more potent, right?
Yes, which mainstream pharmacology and toxicology completely reject because it is just scientifically incoherent. It completely defies the dose-response curve we know governs every single interaction in biochemistry.
So homeopathy ignores the math intentionally. But history is full of moments where mainstream science just got that math wrong by accident.
Very true.
And with catastrophic results. When we fail to respect the therapeutic window, it is a matter of life, death, and sweeping legal reform. Which leads us to the thalidomide tragedy.
Yes. In the late 1950s and early 1960s, thalidomide was aggressively marketed across dozens of countries as this remarkably safe sedative and anti-nausea drug, and it was specifically recommended for pregnant women experiencing morning sickness.
And the really tragic part is that the animal testing data actually backed up those safety claims at the time, right?
It did. And that is where the pharmacology gets intensely complex. Mice and rats, which were the standard laboratory models, metabolize thalidomide differently than humans do.
Oh, I see.
Yeah, their bodies break it down in a way that actually bypasses the severe fetal toxicity. So on paper, based entirely on the animal data, it looked perfectly safe. It was a false sense of security that became the hardest toxicology lesson of the 20th century.
Because humans process it differently, it resulted in over 10,000 children worldwide being born with severe birth defects, particularly phocomelia, which involves malformed or even absent limbs.
It was devastating.
But there is an incredible hero in this story for the United States who prevented that number from being much, much higher. Dr. Francis Oldham Kelsey.
A truly remarkable figure, Dr. Kelsey was a medical officer at the U.S. Food and Drug Administration. The drug manufacturer was putting immense, relentless pressure on the FDA to approve thalidomide for the American market. pointing to its, widespread use in Europe.
But she said no.
She repeatedly refused to sign off. She dug into the data and felt the safety evidence, particularly regarding how it might affect a developing fetus, was simply insufficient.
Wow.
Her scientific stubbornness literally prevented the thalidomide tragedy from taking root in the United States.
And her refusal directly catalyzed a massive shift in how we handle drugs today, didn't it?
Oh, absolutely.
The 1962 Kefauver-Harris Amendment was passed largely because of this near miss. That amendment mandated that drug companies had to prove a drug was not just safe, but also strictly effective in controlled trials before it could ever be approved.
It is the absolute bedrock of modern pharmaceutical regulation.
It is. And these high stakes aren't just historical footnotes. We navigate these exact principles in emergency medicine every single day.
We do.
Like if someone is having an ischemic stroke, where a blood clot is blocking oxygen to the brain, physicians race to administer those thrombolytic drugs we talked about earlier, clot busters like TPA.
Right, because the medical mantra is time is brain.
Exactly. But that drug, which saves brain tissue by physically dissolving the clot, is incredibly dangerous.
Very dangerous. If it is given outside of a very specific time window, usually just a few hours from the onset of symptoms, or given to a patient with a different type of stroke, it can cause catastrophic, fatal bleeding in the brain.
That is the knife's edge, the therapeutic window. And we see the devastating consequences of tolerance shifting that window in the opioid epidemic too, right?
Tragically, yes. The opioid crisis is fundamentally a crisis of pharmacology overlapping with toxicology.
Oh, so.
Well, the exact same molecular mechanism that binds to receptors in the brain to provide profound pain relief is the one that depresses respiration.
Oh, I see.
As a patient's body develops tolerance to the drug, they require a higher and higher dose just to get the same pain relief. But their respiratory system doesn't always build tolerance at the same rate.
So the curves start to separate.
Exactly. Eventually, the dose required for relief pushes them dangerously close to the toxic threshold where they simply stop breathing.
So we see how this math affects an individual patient. But how does this scale up? Like, what about public health and the environment?
This is where we zoom out to population-level toxicology. When you look at systemic crises, like the lead contamination in the water supply of Flint, Michigan, or the ongoing global anxiety about PFAS, the so-called forever chemicals accumulating in our environment.
Right.
Regulators are using these exact same principles. They have to take those dose-response curves and apply them to chronic, low-dose exposures spanning millions of people over decades of their lives.
Which feeds into a really interesting and highly contested debate in the field right now regarding something called hormesis.
Ah, yes. Hormesis is a highly provocative concept. So the traditional model for things like radiation or environmental toxicants is linear no threshold.
Meaning what?
That basically means any amount of a bad thing above zero carries some proportional risk to your health. Just a straight line up.
Okay, but hormesis argues something different.
Right. Hormesis suggests a U-shaped or J-shaped curve. It's the hypothesis that certain toxic substances or extremely low-level radiation might actually trigger a beneficial cellular stress response in the body at microscopic doses.
Wait, really?
Yeah. The substance only becomes harmful once a specific threshold is crossed.
So you're saying a tiny bit of a known toxin might actually kick your immune system into gear and make you stronger before a larger dose inevitably harms you?
That is the argument, yes.
Oh.
It is heavily debated among regulators and toxicologists today, but it perfectly illustrates how incredibly complex the biological interpretation of dose really remains.
It really does fundamentally shift how you look at the world around you. We've covered a massive amount of ground today, but when you zoom out, the through line is undeniable.
It really is.
The fact that the Greek word pharmakon ambiguously meant both drug and poison wasn't just a quirk of ancient language, it was a deeply accurate reflection of biology. And Paracelsus proved that with his realization that the dose alone dictates the outcome. We see that mechanistically when we use a suffix like hemolytic, which simply means dissolving, relying entirely on the context to tell us if a wrecking ball is clearing a dangerous clot or destroying healthy cells.
And we see it in the molecules themselves, whether it is warfarin transitioning from a pesticide to a daily blood thinner, or a botulinum toxin going from a world-ending threat to a targeted cosmetic therapy. The identity of the molecule doesn't decide its morality.
Exactly.
And as the thalidomide tragedy proved, when we misunderstand how a body metabolizes a dose, the consequences alter the course of medical history forever.
If you're anything like me, you are probably going to look at that medicine cabinet a little differently tonight. Because if the dose alone makes the poison, and if modern toxicology technically encompasses everything from lethal venoms to the everyday water and oxygen we need to survive, it means your body isn't just a static, solid object.
Not at all.
You are a real-time, walking, breathing dose-response curve. Every single second of your life, you are perfectly balancing on a biochemical knife's edge, constantly managing and filtering the toxicity of the world around you simply by staying alive. Something to think about the next time you take a deep breath or, open that cabinet under the sink.